Welcome back to the deep dive. You know how this works. You bring us these incredibly dense foundational sources, and we pull out the absolute highest yield concepts and the clinical correlations you just have to know.
That's the goal. Today we're cutting a path straight through biochemistry and genetics.
A rapid path for sure.
Exactly. We're not just memorizing facts. We're trying to follow the flow, from a single nucleotide all the way up to complex metabolic diseases. We're really hunting for the so what of every single step.
Because understanding the why is the key.
It's the only shortcut that works.
And really the whole story today boils down to two things. The flow of information DNA to protein, and the flow of energy that makes it all possible. So let's start at the beginning. The molecular blueprint.
Right. The central dogma. It's the foundational rule. DNA stores the information. RNA is the messenger that carries it. And protein is the machine that gets built.
And the structural difference between DNA and RNA seems so small, but it's everything.
It is. RNA has a 2' hydroxyl group on its sugar. DNA doesn't, which is why we call it deoxyribose. That one little chemical group makes RNA way less stable.
And that directionality, the 5' versus 3' end, why is that so non-negotiable for the cell?
Well, because it dictates how everything is built. You link nucleotides with a phosphodiester bond. It connects the 5' phosphate of a new block to the 3' hydroxyl of the chain that's already there.
So if you don't have that 3' hydroxyl?
The assembly stops, dead in its tracks. That's why polymerization always goes in one direction, 5' to 3'.
And for stability, we have the base pairing, GC being the stronger of the two.
Much stronger. 3 hydrogen bonds between guanine and cytosine versus only 2 for adenine and thymine. And this predictability is key. In double-stranded DNA, A has to equal T and G has to equal C. That's Chargas rule.
Right.
Even RNA, which is single-stranded, will fold back on itself using those same pairing rules to create functional shapes, like hairpin loops.
Okay, so you've built this incredibly long strand of DNA. Now you have, 2 meters of it?
About 2 meters, yeah.
And you have to fit it inside a nucleus that's only 10 micrometers wide. I mean, how is that even physically possible?
It's an amazing packaging system. The most basic unit is something called the nucleosome.
Okay.
You take 146 base pairs of DNA, and you wrap it around this protein core, which is called the histone octamer. These histones, H2A, H2B, and so on, are super rich in positively charged amino acids.
Ah, so the DNA backbone is negatively charged because of all the phosphates.
Exactly.
And the histones are positively charged, so they stick together like magnets.
Precisely. And how tightly they're stuck together is basically the master switch for gene expression. Really tightly packed DNA is this heterochromatin. It's condensed, it's silent.
And the loose stuff.
That's euchromatin. It's open for business, ready to be transcribed.
Yeah.
And then another histone, H1, comes in and links all these nucleosomes together to pack it even tighter.
So let's move on to actually copying all of this.
Yeah.
Replication. That 5' to 3' rule means one strand is easy, the leading strand.
Right, it's continuous.
But the other one, the lagging strand, has to be built backwards in these little pieces.
The Okazaki fragments, yeah, which then have to be stitched together. But what's truly incredible is the accuracy.
The fidelity.
Yes. DNA polymerase, the enzyme doing the copying, makes a mistake about once every 100,000 bases.
That sounds pretty good.
It sounds good
Yeah.
until you do the math. Over the entire human genome, that's 60,000 errors every single time a cell divides. The cell would just fall apart.
So there has to be a quality control check.
There is. And it's built right into the polymerase. It has a 3' to 5' editing function and exonuclease.
So it proofreads its own work.
It does.
Right.
As it's moving forward, 5' to 3', if it senses a mistake, it literally backs up, snips out the wrong base, and tries again.
And that one function?
Improves the error rate 100 times, down to about 1 mistake in every 10 million base pairs.
And that mechanism is something we can exploit clinically, right? With nucleotide analogs.
Yes. This is a huge deal in chemotherapy and antiviral therapy. Drugs like AZT for HIV or cytosine arabinoside for cancer. They are fake nucleotides.
Big how.
They're missing that critical 3' hydroxyl group. So when the polymerase tries to add one to the growing chain,
The chain stops.
it just halts. It's a very targeted way to stop cells from replicating.
Speaking of stopping, replication has a physical problem at the very end of the chromosomes.
Right, the end of the line problem. The machinery can't copy the very tips, so the chromosomes get a little shorter with every division.
And those tips are the telomeres.
Exactly. They're just repeating sequences, TTTG,
Yeah.
that act as a buffer to protect the actual genes. But eventually they run out and the cell stops dividing,
Unless.
unless you have an enzyme called telomerase. It's a reverse transcriptase that can rebuild those telomere ends, which basically confers a form of cellular immortality.
Which is great for stem cells, but a huge problem in cancer.
A huge problem. Most cancer cells find a way to turn telomerase back on, which is why they can divide indefinitely. And it's why telomerase inhibitors are such a hot area of research.
What about damage from the outside world, like UV light?
UV light causes a very specific type of damage. It fuses adjacent pyrimidines together, usually two thymines, creating what's called a dimer.
And the cell has to fix that.
It has a dedicated system. An endonuclease recognizes the bulge in the DNA, snips out the damaged section, and then DNA polymerase fills in the gap. It's a constant process of surveillance and repair.
So let's move from copying the information to actually using it, transcription.
Right, going from DNA to RNA, using RNA polymerase. And a great clinical tie-in here is the antibiotic rifampin. It works by blocking that very enzyme.
And in our genes, we have the parts that code for protein, the exons.
And the parts that don't, the introns.
Both get transcribed, but then the introns have to be removed.
They do. And the whole process is regulated at multiple levels. You have general promoters, like the TATA box, that say transcribe here. But for a gene to be expressed at high levels, you need enhancers.
And enhancers are weird, right? They can be really far away from the gene.
Thousands of bases away. They can even be inside an intron. The DNA literally loops around so the enhancer can touch the promoter and crank up transcription.
And all of this is layered on top of epigenetics.
Yes, things like DNA methylation or histone deacetylation. As a general rule, those things tighten up the chromatin and silence genes.
Okay, so after you make the raw RNA transcript, you have to process it. A cap on the 5' end, a tail on the 3' end.
A poly A tail, yeah. Both protected from being degraded.
And then the big one, splicing. How does the cell know exactly where to cut out the introns?
It uses a huge molecular machine called the spliceosome. It recognizes specific sequences at the start and end of an intron, usually a GU and an AG, and it cuts the intron out in this unique looped structure.
A lariat.
A lariat intermediate, exactly. It's like a molecular lasso that ropes the intron and pulls it out, then perfectly stitches the two exons together.
And what's wild is that the cell can splice the same gene in different ways.
Oh, absolutely. It's called alternative splicing, and something like half of all human genes do it. It's how one gene can produce multiple different proteins.
Like an antibody that can be either stuck to a cell or secreted.
That's the classic example. You get a transmembrane version and a secreted version from the same gene just by splicing it differently. It's incredibly efficient.
Okay, let's get to translation. Making the protein, you need the processed mRNA, ribosomes, amino acids, energy.
And all the different factors. And the way those amino acids are strung together matters. You have a distinct N-terminus and C-terminus.
And the properties of the amino acids themselves determine the final shape.
For sure. The hydrophobic ones, the aliphatic ones, they bury themselves on the inside of the protein away from water. The polar and charged ones stay on the surface.
And the key middleman here is the tRNA, which has to be loaded with the correct amino acid.
By an enzyme called minoacyl tRNA synthetase. And that enzyme is another critical checkpoint. It has its own proofreading function to make sure it's attaching the right amino acid.
Once the protein is made, it doesn't just fold up on its own, does it?
Not always. It often needs help from chaperone proteins. You can think of them as the quality control inspectors. If a protein is misfolded, like the one in cystic fibrosis, the chaperones might tag it for destruction.
And the cell's garbage disposal is the proteasome.
It is. A protein that's destined for destruction gets tagged with a chain of another small protein called ubiquitin. It's a kiss of death tag. The proteasome sees the tag and just shreds the protein.
Let's talk about sending proteins to the right place, specifically lysosomal enzymes.
This is such an elegant system. These enzymes are made in the RER, they get some sugars attached, but the key step happens in the Golgi. An enzyme phosphorylates one of those sugars, a mannose.
So it adds a phosphate group.
It does. And that mannose 6-phosphate is like a zip code. It's the address label that says, send this package to the lysosome.
So what happens if the enzyme that writes the zip code is broken?
Then you get a devastating condition called eye cell disease. The phosphotransferase is defective. The lysosomal enzymes never get their address label.
So they don't go to the lysosome.
They get secreted outside the cell instead. And meanwhile, the lysosomes inside the cell fill up with junk they can't digest. It's a catastrophic failure of cellular logistics.
Let's touch on one last structural protein, collagen. It's so high yield because its synthesis is so complex.
It really is. Its primary structure is defined by having a glycine at every third position. That lets it form this unique left-handed helix.
And then 3 of those wrap around each other.
into a right-handed triple helix,
Yeah.
a massive, strong rope. And it's stabilized by modifying proline and lysine into hydroxyproline and hydroxylysine.
Which requires vitamin C.
Absolutely. Requires vitamin C as a cofactor. That hydroxylation step happens inside the cell. If you don't have vitamin C, you can't make stable collagen.
And that's scurvy.
That's scurvy, bleeding gums, poor wound healing. And then you have genetic diseases like osteogenesis imperfecta or Ehlers-Danlos, where the collagen protein itself is built wrong from the start.
Okay, huge shift now. Let's talk about energy, glucose metabolism.
We have to start with how glucose even gets into the cell, through GLUT transporters. GLUT4 is the famous one in muscle and fat that responds to insulin.
But the one in the liver, GLUT2, is different.
Very different. It has a high CAM, which means it has a low affinity for glucose. That sounds bad, but it's brilliant. It means the liver only starts taking up large amounts of glucose when blood sugar is really high, like after a meal. It leaves plenty for the rest of the body.
Once inside, we have glycolysis. 2 ATP, 2 NADH. But the key regulatory point isn't the first step, it's PFK1.
Phosphofructokinase 1. It's the main control valve. If the cell has plenty of energy, high ATP levels will inhibit it. But the main activator is a molecule called fructose 2006-bisphosphate, whose levels are controlled by insulin and glucagon.
The end product is pyruvate, which can go a few different ways. What happens if the enzyme that sends it into the aerobic pathway, pyruvate dehydrogenase, is broken?
You get pyruvate dehydrogenase deficiency. The pyruvate has nowhere to go but to lactate, so it builds up, causing a severe chronic lactic acidosis.
And the treatment is very specific.
You have to limit the fuel source, so a high-fat, low-carb diet. And you give high doses of thiamine because it's a critical cofactor for that enzyme.
Now, contrast that with hereditary fructose intolerance.
This is a deficiency in aldolase B. When someone with this eats fructose, a molecule called fructose 1-phosphate builds up. And this molecule is a phosphate trap. It sequesters all the free phosphate in the cell.
And if you don't have free phosphate,
You can't make ATP. And without ATP, you can't do gluconeogenesis or break down glycogen. So you get profound, dangerous hypoglycemia after eating fructose.
let's talk about that lactate pathway for a second. In red blood cells, which don't have mitochondria, what is the whole point of making lactate?
It's all about regenerating NAD+1. Glycolysis needs NAD+ to run. The conversion of pyruvate to lactate takes NADH and turns it back into NAD+1.
So it's a way to keep glycolysis going just to make those 2 little ATPs.
Exactly. It's their only source of energy. It's a lifeline. The liver can then take that lactate and turn it back into glucose later in the Cori cycle.
That balance between NAD+ and NADH is also what gets completely wrecked by alcohol metabolism.
Completely. The breakdown of alcohol generates a massive amount of NADH. The NAD plus NADH ratio plummets. This flood of NADH pushes pyruvate to lactate and basically shuts down gluconeogenesis.
Which leads to alcoholic hypoglycemia.
Right, and this is why the clinical rule is so important. If you have a patient you suspect is an alcoholic and hypoglycemic, you must give thiamine before you give glucose.
Why?
Because the glucose will push the remaining metabolic pathways into overdrive, which requires thiamine as a cofactor. If they're already deficient, you can precipitate a severe neurological condition called Wernicke-Korsakoff syndrome.
Last metabolic pathway, the HMP shunt. It runs alongside glycolysis.
Its main jobs are to make 2 things. Webos 5-phosphate for building nucleotides,
And NADPH.
and NADPH. The cell's primary reducing power. You need it for making fatty acids, cholesterol, and most importantly, for regenerating reduced glutathione to fight oxidative stress.
Which brings us to G6PD deficiency.
The most common human enzyme deficiency. It's X-linked. Red blood cells are uniquely vulnerable because the HMP shunt is their only source of NADPH. Without it, they have no defense against oxidative damage and they just lose, causing hemolytic anemia.
Okay, let's pivot to genetics. X-linked recessive diseases are way more common in males.
Because they're hemizygous. They only have 1 X. Females have 2, and 1 gets randomly inactivated in each cell, forming a bar body. They're mosaics.
Sometimes that inactivation isn't so random.
Right. If it's skewed, a female carrier might mostly inactivate her healthy X chromosome, and she could actually show symptoms of the disease. We call that a manifesting heterozygote.
Let's untangle a few complex patterns, one gene causing many different problems.
That's pleiotropy. Marfan syndrome is the classic case. One bad fibrillin gene causes problems in the skeleton, the eyes, the heart.
And a disease that gets worse with each generation.
That's anticipation. It's usually caused by trinucleotide repeat expansions, like the CAG repeats in Huntington disease. The repeat gets longer in successive generations, and the disease starts earlier.
And then there's imprinting.
Imprinting is fascinating. It's where a gene is silenced based on whether it came from your mother or your father. For certain genes, you only have one working copy. If that one copy gets deleted, you have a problem.
Like with Prader-Willi and Angelman syndromes.
Both on chromosome 15. It just depends on whether the deleted copy was the paternal one or the maternal one. You can also get these diseases from uniparental disomy, where you inherit both chromosomes from the same parent.
Finally, let's look at the big picture. Cytogenetics and aneuploidies.
This is when you have the wrong number of chromosomes, usually from non-disjunction, a failure of chromosomes to separate properly during meiosis.
The three major viable trisomies are
Trisomy 21, Down syndrome, trisomy 18, Edwards syndrome, and trisomy 13, Patau syndrome. Edwards and Patau are much more severe, with most infants not surviving the first year.
and for the sex chromosomes.
You can have Turner syndrome, which is 45X. It's the only viable monosomy in humans. And Klinefelter syndrome, which is 47,000XXY.
That was an incredible tour. We've gone from a single hydroxyl group all the way to entire chromosomal abnormalities and seen how it all links back to regulation and energy.
It really hammers home that none of these pathways exist in a vacuum. The cell's health depends entirely on the real-time balance of all these moving parts, especially cofactors like vitamins and energy carriers like NAD plus CODs. One little block can cause a system-wide crisis.
Absolutely. So to help you synthesize all this, especially the metabolism and disease sections, here is your challenge question for this deep dive. We discussed two diseases that result from a block early in carbohydrate metabolism. Contrast the pathophysiology of pyruvate dehydrogenase deficiency and hereditary fructose intolerance. Think about the deficient enzyme, what toxic substance accumulates, and why that leads to the primary clinical symptom.
Right. Think about where the block is in each case. The accumulated molecule is what causes all the downstream damage, but the damage is very different. One is an acute acid problem, the other is an acute energy failure.
Keep synthesizing this high-yield knowledge. We'll see you on the next deep dive.